So for this course, I had to pick an article to read and study throughout the remainder of the course. I chose Early Life Stress Can Disrupt Maturation of Brain's Reward Circuits, Promoting Disorders. Of course, right? Verbatim. In an article recently published online in Nature Communications, Dr. Tali Z. Barham, senior author and UCI Donald Bren professor and distinguished professor in the departments of anatomy and neurobiology, pediatrics, neurology and physiology, and biophysics, and Matt Burney, lead author and postdoctoral researcher, describe the cellular changes in the brain circuitry caused by exposure to adversity during childhood. We know that early life stress impacts the brain, but until now, we didn't know how, Barham said. Our team focused on identifying potentially stress-sensitive brain pathways. We discovered a new pathway within the reward circuit that expresses a molecule called corticotropin releasing hormone that controls our responses to stress. We found that adverse experiences cause this brain pathway to be overactive. These changes to the pathway disrupt reward behaviors, reducing pleasure and motivation for fun, food, and sex cues in mice, she said. In humans, such behavioral changes called anhedonia are associated with emotional disorders. Importantly, we discovered that when we silence this pathway using modern technology, we restore the brain's normal reward behaviors. Researchers mapped all the CRH expressing connections to the nucleus accumbens, a pleasure and motivation hub in the brain, and found a previously unknown projection arising from the basolateral amygdala. In addition to CRH, projection fibers co-expressed gamma-aminobutyric acid. They found that this new pathway, when stimulated, suppresses several types of reward behaviors in male mice. The study involved two groups of male and female mice. One was exposed to adversity early in life by living for a week in cages with limited bedding and nesting material, and the other was reared in typical cages. As adults, the early adversity experiencing male mice had little interest in sweet foods or sex cues compared to the typically reared mice. In contrast, adversity experiencing females craved rich, sweet food. Inhibiting the pathway restored normal reward behaviors in males, yet it had no effect in females. We believe that our findings provide breakthrough insights into the impact of early life adversity on brain development and specifically on control of reward behaviors that underlie many emotional disorders. Our discovery of the previously unknown circuit function of the basolateral amygdala nucleus accumbens brain pathway deepens our understanding of this complex mechanism and identifies a significant new therapeutic target, Barham said. Future studies are needed to increase our understanding of the different and sex specific effects of early life adversity on behavior. The part where it said, as adults, the early adversity experiencing male mice had little interest in sweet foods or sex cues compared to typically reared mice. In contrast, adversity experiencing females craved rich, sweet foods. Isn't that crazy? Do you see how that is so similar to us humans? Throughout adversity, the male mice had no drive, no ambition, no hunger. Whereas the females, the women that was in this same environment of adverse effects, they still crave rich and sweet foods. Doesn't that sound familiar? Don't a lot of females still want a lot of things they don't have?